Hello there. In this video, we're going to continue our exploration of the concept of poisons by first talking about what a poison is in the first place. Um, I'd like to draw your mind to the opening scene in Hocus Pocus, where the three Sanderson sisters are standing around the cauldron and they're just throwing in all sorts of different seemingly benign ingredients, things like a frog, a dead man's toe, a hair, saliva from their own mouths. And so all of these things, unlike the scene in Macbeth from the last video, all of these things wouldn't necessarily seem to be poisons first, when you first bring this to mind, but in fact, they ultimately produce this deadly poison that will steal the soul and the life of this little girl. And so on the screen, you see a quote or translation of a quote in German by Paracelsus. In the 1500s, he says, "All things are poisons, for there is nothing without poisonous qualities," or some translation therein. And so the Sanderson sisters mixing all of these different ingredients together alone, they might not have been considered poisons, but in fact, they do have these poisonous qualities. And the second half of this quote, which is to this day debated, but praised by many as the central concept in toxicology, and that is essentially the dose makes the poison. And so we're going to explore that a little bit in this video, as well as throughout this series. So a little bit more about Paracelsus. Paracelsus is not his given name. As you can see on the screen, his given name was much longer, but he named himself Paracelsus because he was kind of an arrogant, guy, very stubborn, and he claimed that he was, that he was so intelligent, that he was really onto something so much more than his contemporaries of his day, that he was even above Celsus, uh, you know, a, a, uh, a doctor essentially in the first century CE in ancient Rome. And so let me take a step back here. Paracelsus was born again 1493. He grew up in a mining town, essentially at his father's side. His father was essentially the equivalent of a physician at the time. Of course, this was the beginning of the Renaissance, where we have this shift from just women and their herbal remedies passed down from generation to generation, just healing individuals in their household and others in the community with herbal remedies and other types of things, and essentially, a lot of different individuals within the community would have this kind of knowledge, but at this time, you start to see a shift from just rural communities into more urban environments. And so that information in that role was no longer adequate to support the growing population of the time. And so you started to see individuals that would specialize in this evolving practice of medicine. And so I say that his father was a physician, but in fact, he was just like the town healer in this mining town. And so, uh, you know, very early on, Paracelsus had this connection between essentially occupational hazards, right? What these individuals in the mines are being exposed to, not only from debris and whatnot, but from uh, particulates in the air that they're breathing in from exposure to heavy metals. And so Paracelsus would have seen a lot of disease related back to chemicals. And so this connection between chemistry and health was at the forefront of his young mind. And so, as I said, Paracelsus was quite the character. At 14, he just left home and started wandering around Europe and a little bit into Asia and Africa, essentially seeking knowledge, which was very noble thing during the Renaissance. Essentially, he went to different universities and tried to just soak up as much information as he possibly could. Specifically about chemistry and about medicine. He did spend some time in the army as a surgeon and apprenticing with surgeons as well. And he was a self-declared alchemist. Ultimately, he was invited to be a speaker, a professor of medicine at the University of Basel, as well as the city physician. Again, this wasn't a physician like we would think of today. There was no medical school. Specifically, there were no degrees necessarily, but he was trusted with this healing capacity. Keep in mind that at this time, even stitches as opposed to cauterization was a really new idea. And it wasn't a physician's job necessarily at the time to do those types of techniques. At this point, barbers were the ones who were responsible for stitches in patients. Anyway, this was a really prestigious role to be invited into both of these professions. But during his time in these roles, he spoke often and loudly about his very controversial at the time views on medicine, and of course, as part of that, his views on poison. He he had a lot of different controversial views, but the ones that are relevant to this talk are this thought that chemistry was important to medicine, that disease wasn't just a reflection of sinning and God's punishment of the sinner. It wasn't even about the four humors being out of line. Instead, there's this strong connection between chemicals and health. And so he said that he pioneered the use of minerals and inorganic compounds in medicine, but keep in mind that this wasn't his idea. In fact, hundreds of thousands of years before, inorganic compounds and heavy metals, etc., were used in medicine. And he merely reintroduced this concept back into the medical field of his time. Also, I have in parentheses here European, because in other parts of the world, this was always the case, much more continuous use of this concept. And so because of his experiences, he ultimately was a big fan of using things like mercury and arsenic, which today we would very much consider to be poisons, but he would use them in tinctures to help heal individuals as well. And some people actually were healed by these treatments, such as Louis XIV was healed by antimony. And so one of his big take-home messages was that plants are out, herbal remedies are just not sufficient. Chemicals are really what can heal severe illnesses. And so ultimately, he put forth that like cures like, a poison in the body would be cured by a similar poison. And so, this of course ties back to the dose makes the poison. And so Paracelsus, again, was not popular in his day. He would kind of attack people that opposed his views, which was pretty much everyone at the time, and his demonstrations of his views just became more and more aggressive, kind of culminating in this display at a St. John's Day bonfire, where he literally threw the book of medicine at the time into the bonfire. It was just like, nope, this stuff is is not true. Okay, so during his life, he was very, very unpopular. It wasn't until decades after he died that that people started to realize that, oh, like he was kind of onto something. To this day, his work is controversial. There are many papers in the literature that start with the dose makes the poison. Yes, that is the case. We are going to support that here, or the dose makes the poison. This is a load of crap, and we're going to tell you why here. And so, still to this day, this guy is making people talk. Okay, so let's talk about the current definitions of poisons. If you simply Google what a poison is, a lot of definitions really focus on the negative aspect, on the harm that poisons can cause. But in this series, I am really going to try to show how poisons can have both good and bad effects, depending on, of course, the dose, as well as some other things as well. So the definitions that we're going to be looking at are here, one from a veterinary textbook, the other from a medicine or forensic medicine and toxicology department. And so both of these definitions highlight is that something is coming from the outside of the body and it's going to interfere with whatever physiological process is occurring naturally within the body. Okay, so this can literally be anything, like you take a bite of a hamburger, that is something from the outside of your body, it is coming into your body, and it is going to change life processes that are already occurring. Right? And so this, of course, would generally be a good thing. It is going to fuel different metabolic reactions and so on. But it is changing the life processes occurring within your body. The other key part of both of these definitions is that the molecules themselves are going to disrupt biological processes by their inherent properties. They're not, they don't need to be modified before they start acting. Okay. And so just some examples here, very basic. In our nervous system, so in our brain, our spinal cord, our nerves, essentially the functional cells are, they're called neurons. Here we can see neurons. Essentially, they're going to relay messages from one part of the body or the brain or whatever to another part and essentially produce some sort of downstream pre-programmed effect. And so the way that they are able to relay these messages is by releasing little chemical messengers called neurotransmitters. Here we see the end of one neuron, here we see the beginning of another neuron. And so to communicate back and forth, what's called presynaptic neuron releases these little neurotransmitters. You've probably heard of some things like dopamine, serotonin, acetylcholine. Chemical messengers, lots of different types, and they relay different messages depending on what they are and where they are. And so ultimately, poisons can disrupt a life process, like relaying messages from the brain to, for example, your skeletal muscles so that you can clench your fist, for example, by disrupting this communication process here. So some ways that this could happen is, maybe blocking the receptors on the downstream or postsynaptic cell. So even if you are thinking like, okay, clench my fist, clench my fist, clench my fist, that message isn't actually going to be able to get to your muscles because, even though these messengers are being sent, the receptors, right, essentially the ears listening for this message are no longer able to function. Another way that this life process could be interfered with is maybe blocking the synthesis of these neurotransmitters, maybe blocking their ability to be released, okay, kind of plugging up these channels here, which ultimately allows the neurotransmitters to be released. And so regardless of the mechanism, the effect of this poison, or also called toxicant, is that you can be thinking, clench my fist, and nothing happens. That message doesn't get there. And so what that would result in is paralysis. Another example is carbon monoxide, an odorless gas, which is one carbon, one oxygen. Essentially, when you breathe in carbon monoxide, it is going to bind to the proteins within your red blood cells that are responsible for carrying oxygen. right? So these hemoglobin molecules are important because they are actually going to deliver oxygen from your lungs to the rest of your tissues. And of course, your tissues, right, your skeletal muscles, your neurons, all of these different things require oxygen in order to function. And so when carbon monoxide is inhaled, it is going to bind to those hemoglobin molecules within your red blood cells, your erythrocytes, and it is going to essentially plug up the hole that is usually used to carry that oxygen. And so instead of carrying oxygen, you're carrying carbon monoxide. Your body cannot use carbon monoxide. And so your tissues essentially starve of oxygen. And so you end up with hypoxia as a result of inhaling carbon monoxide. So those are just a couple examples. We'll talk a lot more about different mechanisms of how exactly poisons, or also called toxicants, work in the next video. Last thing I want to mention about poison as a term is that this can refer to a biological poison. So something like, like what is released in a bee sting, or it can be anthropogenic. Right, so maybe we made some kind of poison on purpose or not. It could also refer to heavy metals and such, right? So different inorganic substances already in the environment. Okay. Within this blanket term of poison, we can also talk about toxins, also called biotoxins. These are poisons that specifically originate from biological processes. Now, I do want to make the distinction between pathogen or parasite and toxin. Toxins are generally chemicals, and if the entire organism itself is toxic, it disrupts biological processes in your body, then we would call the organism a pathogen or a parasite, depending. Okay. Also, within this blanket of toxins, right? So poisons, toxins, within toxins, we can also classify these chemicals based on their route of entry. Okay, so for example, if a poison must be injected subcutaneously under the skin in order to function, it is called a venom. Okay, so what we can see here is a bee sting. And so what is happening here is that the venom is injected under the skin. It is more or less immediately taken into the blood and taken into the lymphatic circulation and ultimately distributed throughout the entire body. When this substance, right, these molecules, usually proteins, are injected, there are fewer, or I guess just kind of different defense mechanisms to counteract what is happening with this poison. As opposed to if you were to ingest bee venom or snake venom or something. If you ingested these molecules, essentially when you swallow that substance, it ends up in your stomach. And as you know, the stomach has lots of stomach acid. The thing about stomach acid is that it denatures or unravels proteins. And so if the bee poison is proteins, right, which it is, essentially those proteins would be unraveled in the stomach and therefore no longer functional, no longer able to disrupt our normal biological processes. And so one of the reasons that your stomach does have acid is because it's kind of acting as a defense mechanism against poisons. The other thing is that your GI tract, gastrointestinal tract lining, has a lot of different mechanisms to prevent the poison or anything else from actually getting into your blood, right? Once it's in your blood, it's in your body and it's wreaking havoc. But if we can keep these things out of our blood, then we can eliminate them without allowing them to hurt us. Right, so here we can see the lining of the stomach. All of these cells here, these epithelial cells, are joined together by what are called tight junctions. They make sure that the membrane itself is solid and doesn't allow anything to slip through it, sorry, and ultimately like squeeze between these cells and get into the blood. The stomach also has a layer of mucus and some other things that really prevent poisons from getting into the blood. And of course, plenty of poisons do get beyond these barriers and into the blood and wreak the havoc that they do. But here I just want to make the distinction that venom is generally made of proteins, and therefore, in order to function as a protein or as a poison, it needs to be injected under the skin where there aren't those protective mechanisms like acid, tight junctions, etc. Toxic. Okay. Toxic definition is essentially the adjective form. It is describing these poisons or these toxins. Originally, it was supposed to just refer to the action of a toxin, so something made by a biological organism, but in fact, this is at this point used to describe either a toxin or any other kind of poison. Okay. And usually toxic is used interchangeably with poisonous at this point. Do you want to mention that this word is derived from the Greek word that means bow. And so just like I talked about in the last video with Hercules, where there were, a lot of interesting feelings by the Greeks reflected in this mythological tale between weapons and poisons and the danger of the two together. And, not only are they lethal as intended, but there are also a lot of, side effects, a lot of unintended consequences. And so literally the word for bow is connected to toxic, right? Because these individuals were using these biological weapons at the time. Also, there's a connection with another root word in Greek, and that is taxus. The genus of the yew tree, which is this right here, is Taxus, and this is further connected to toxic and toxin because the yew tree was actually used for making bows, right? So you can see the connection immediately there. So also, not only was the wood valuable for bows, but the seeds have poison, right? The seeds and the berries that surround them. And so the yew tree was very important to make these biological weapons. Historically, these plants were planted in churchyards, especially during the Dark Ages, in order to protect animals from the poisonous berries. Another term for you, toxicity. This is describing the capacity of a chemical to produce injury. This specifically is referring to detrimental biological changes, right? So yes, poisons can be used to heal, but also they can be used to harm, especially, or that they can also be used to harm. Right, so toxicity is specifically talking about the detrimental biological changes produced by these poisons. Also, toxicity levels aren't just, we can't describe toxicity in a single number. There's always some kind of extra information that is needed. So toxicity under a specific set of conditions. And so what this implies is that toxicity is affected by a variety of different factors. And so I'm going to walk through a few of them here. First of all, the species. So for example, chocolate is toxic, so high toxicity level in dogs, but lucky for us, not so much in humans, right? So the species is really affecting the toxicity of the same substance. Right. In addition to the species, the species itself, a lot of times the differences in toxicity is related to size. So we can see here that the rock is a very different size than his little puppy here. But of course, there are other humans that are a very different size than canines, right? And so this isn't, you know, always the case. There's a lot of variability even within species. Um, so a lot of variability within species. So for example, the rock has a lot bigger mass, a lot larger mass, as opposed to this little girl here. And so eating the same um, volume, or being exposed to the same volume of a poison might affect the rock versus this little girl very differently. So for example, if um, the dose, so the amount of toxicin received is a full-size Snickers bar, right? That full-size Snickers bar per 20 pounds of this little girl versus, however much the rock weighs, that dosage, right, the amount of toxicin versus the body weight, is going to be very different. Okay. Similarly, right, Great Dane versus Frenchie here, very different dosage, even if they're eating the same chocolate bar. Okay. Furthermore, species vary, or toxicity varies between species because simply their genetics are different. So toxicity is higher for chocolate in dogs because they are unable to process, break down, metabolize the chemicals in chocolate like we are, right? We can very easily break those molecules down, and of course very quickly, so that that chemical is excreted from our body pretty quickly. Right. Also within species, genetics is very important. Some people can process poisons very efficiently, others cannot, and they're affected a lot more. Furthermore, age and sex. So first of all, age. All right, this little girl might be affected more by poisons than the rock would be because she is still developing, right? So a lot of her resources within her body are being dumped into growing and, signaling where neurons are supposed to grow, and, making tons and tons and tons of red blood cells. And so lots of her energy, lots of her resources is already being spent on other processes, and there's not as much left over to process poisons. Also, the fact that she is still developing means that poisons can have a lot longer term effects, because they can disrupt the process of development and ultimately, produce things like, abnormalities in the development of your brain. Okay. In other species, age can be very important. For example, embryos can be very much affected by poisons, more so than an adult animal would be. So for example, in eggs, there's a lot of fatty substances that are deposited to essentially support that developing embryo. A lot of poisons are hydrophobic, or they really like to be in lipids and fats. And so when the animal lays the egg, it's also laying a lot of chemicals in that egg as well. And so there's going to be very high, not only dose, but dosage, right? So a lot of chemical per very small animal weight. Okay. Let's see, sex as well can very much affect the toxicity of different chemicals. So for example, again, a lot of poisons are hydrophobic, so they are stored in fat. And so individuals with more fat content have a higher storage capacity for poisons, and therefore the poisons stick around in their bodies longer. And so they are going to be disrupting biological processes for a lot longer. And so their effects can be cumulative, and they can kind of add up as well, which I'll talk about here in a couple slides. And so I bring this up when I'm talking about sex because on average, females are supposed to have a higher fat content than males, and therefore they are going to accumulate more of these poisons. And so the dose gets higher and higher and higher over the course of their lives. And even beyond that, just the location of where the fat is can have implications as well. So for example, in females in particular, breast tissue is made up of a lot of adipose, or a lot of fat tissue. Again, poisons accumulate in fat. And so in breast tissue in particular, this is where lots and lots of poisons are accumulating. And so they can be mutating DNA and disrupting other processes. And so this can increase the chances of things like breast cancer because of exposure to poisons and their storage in that particular tissue. Going back to age here, because so many poisons are stored in breast tissue, and adipose within breast tissue is broken down to provide nutrients for offspring, that is breastfeeding. Essentially, these chemicals can end up in the breast milk and be fed directly to the baby. And so again, this is where age comes in. Younger individuals who are still developing, can experience more detrimental biological changes because they're getting, a high dosage of these chemicals via the breast milk. Right. Some other examples, lead, for example, is stored in the bones. And so anyone who is kind of breaking down or building up bone pretty quickly is at risk of accumulating more lead and therefore experiencing more of these detrimental biological changes. So for example, if you are going through menopause or you are pregnant, your bones are being broken down and built up at a different rate. So that can very much affect the toxicity of things like lead. Also, if you are, suddenly going to be a bodybuilder, right? And you are going to the gym a lot, your bones actually are going to get thicker and stronger to support that extra body mass and that extra strain on your bones because of lifting all of those weights. And so your bones are actually going to build up a lot more, therefore storing a lot more lead. Okay. Another factor that influences toxicity is the environment in which you live. So here we can see a tadpole that is metamorphosing into a frog. And so I bring up this example because when the frog is in its larval stage, so a tadpole, it is aquatic, and therefore any chemicals that are in the water, this animal is being exposed. And so their skin is super permeable when they're in water. And so all of those chemicals are just kind of leaching into the body of the tadpole. As opposed to, when it emerges from the water with legs, it's a frog, living in the air, it might not be exposed to as many chemicals as it was when it was younger in the water. Furthermore, any kind of environmental stressors can increase the toxicity of chemicals. So for example, if there is overcrowding, there's a lack of lack of food, a lack of water. This can increase the toxicity because the animal is already stressed out or is malnourished, and that is going to reduce the resources available to process these chemicals. Okay. See, things like high salinity and low pH can also increase the toxicity of things like heavy metals. So, for example, in the case of pH, acid rain and increased carbon dioxide in the air because of factory and vehicle emissions, for example, that is actually decreasing the pH of our oceans. Again, with a decreased pH, the toxicity of heavy metals is going to increase for marine animals. Very rarely are organisms exposed to a single chemical at a time. Usually we are encountering many different chemicals all at once, and they're going to be interacting with each other and, of course, with our bodies. And so this chart shows some of the different ways that chemicals can interact with one another. So additivity, so when you're exposed to chemical A, it's going to exert this much of a negative effect. When you're exposed also, or when you're exposed just to chemical B, it exerts this much of a negative effect. But when they are together, now you're getting the negative effects from both. Sometimes when you're exposed to just chemical A, right, this effect, just chemical B, this effect, but sometimes one can actually antagonize, it can negate the effects of the other. And so when both are, when you're exposed to both at the same time, sometimes they kind of cancel each other out. And finally, what seems to be more common and probably more scary is synergism and potentiation. Sometimes terms used interchangeably, but essentially potentiation, exposure to both chemicals at once. Essentially, one chemical is going to activate the other chemical. Okay. But of course, what is more common, because everything is a poison, there's nothing without poisonous qualities, is synergism. Right, so chemical A has this much effect, chemical B has this much effect, but when they are when you're exposed to them together, they have a much greater effect than the sum of the two individual chemicals. So just one example here. This is taken from Kim et al. 2021. Essentially, what these scientists did was they exposed cells and cell culture to just ibuprofen, so a non-steroidal anti-inflammatory drug, something like aspirin. And in some trials, they also exposed to alcohol. So a little bit of alcohol, much more alcohol, and different levels of ibuprofen. And so what they measured was cell viability, how many of the cells were still alive at the end of this exposure. And so in summary here, what they saw was that when they, when the cells were exposed to both ibuprofen as well as alcohol, there was a lot more cell death, right? So cell viability has gone down. Down here, we can see that, these cells have died off when they were exposed to ethanol. And of course, all of these red arrows here show how many more cells died when they were exposed to ibuprofen and more alcohol. Okay, so essentially, this is a synergistic effect, right? So just ibuprofen, not very toxic, in fact, very helpful. People take it for a lot of different reasons. Probably just alcohol, also not very toxic, but when you take them together, that is when you start experiencing problems. And so in a human, what this means is that, first of all, if you're taking ibuprofen, don't drink alcohol. What happens here is that, right, the cells in your stomach can start to die off, just like we are seeing here in these in this cell culture. And so if the cells in your stomach lining start to die off, you can't replace them quickly enough, and therefore you start to get little holes or like thinner membranes in certain parts of your stomach. Your stomach acid can then start like passing through those holes and like eating away at cells within your stomach lining. And so ultimately you can get stomach ulcers by taking ibuprofen and drinking alcohol at the same time. And there are other side effects as well, but that one is the most relevant to this data right here. So read the labels on your medicines and don't drink if it says don't drink. Okay. Another factor that influences toxicity is the route of exposure. All right, first of all, you can be exposed through your skin. So, just like brushing up against something, spilling a poison on your skin. In general, the outer layer of your skin, called your epidermis, is 0.5 millimeter to 1.5 millimeters thick. Most of this layer is actually dead cells. So even if you are exposed to this poison, a lot of the tissue that's being exposed is already dead. So it's really not affecting you quite that much. Of course, some poisons can seep through that epidermis, but it has to pass through this much tissue before it actually gets into your blood and therefore into your body to start wreaking havoc. If you ingest a poison, like I mentioned before with the stomach lining, there are a lot of um, a lot of protective measures in place to protect you from poisons when you ingest them. Of course, the stomach acid, there are lots of enzymes to break down poisons if you ingest them. And of course, the lining of your gut is in many places even thicker than your skin. So a poison would have to pass through this much tissue before it gets in your blood and therefore starts wreaking havoc. So skin or dermal exposure, ingestion, oral exposure, or you can inhale a poison. And so when you inhale a poison, essentially the poison isn't filtered out. It's not stopped. Like there's no filter between the outside world and all the way down here in your lungs where the air actually ends up. And so whatever you inhale can end up way down here. And so if we look at the very ends of our bronchial tree, we see these little structures here, which are called alveoli. This is where the air ends up, and lightning fast, air has to go from inside these little spheres, cross over a membrane, and jump into the blood. All right, so as quick as it takes to inhale, exhale, right, in a second, oxygen has to load into the blood and carbon dioxide has to unload out of the blood. And so our lungs are designed to make this process really quick, really seamless. We don't want to have to like stop and like hold our breath, allowing carbon dioxide and oxygen to do its thing, and then finally exhale. We need it to be super fast. And so these membranes within our alveoli aren't this thick, right, which seems really thin now. In fact, they are much, much thinner. So, here's the skin, here's the gut lining. Here, this tiny little spot is the thickness of the alveolar membrane. Okay, so this is generally measured in micrometers, right? So 1,000 times smaller than millimeters. And so the alveolar membrane in our lungs, the barrier between the outside world and our blood, where the poisons can get in and start wreaking havoc, is this thick. All right, just for some perspective, copy paper is 400 to 5,000 times thicker than the lining of your lungs. So really not a thick membrane at all. Generally, inhaled toxicants are the most dangerous because there's virtually no barrier from the outside world to the inside world. Right. Finally, the duration of exposure. Are you exposed once or in a single day? Are you exposed over about a month or less? Are you exposed for a couple months? Or are you exposed for many months or years? This here is poison ivy, by the way. Note the leaves of 3, let it be. So toxicology is the study of all of these adverse effects, essentially examining the toxicity of different substances that you're exposed to. How do we know, right? How do we actually study toxicology? Well, initially it was just trial and error, right? Bob eats some berries, you wait a little while before you eat the berries as well to make sure that Bob doesn't die. Okay? Um, Individuals who are particularly interested in this kind of thing experimented on themselves a lot. And so in a future video, I'm going to talk all about some individuals, such as Cleopatra, who experimented with poisons to figure out just how much was too much. Also, individuals who were so slaves and servants were often required to test, for example, wine before royalty ate or drank, where their food or drink is, or, just more general trial and error with individuals that are in your servitude. Um, and animals have been used quite a lot over the course of history. For example, a reading that we're going to look at in a future video expresses how individuals, if they ate something and they started to vomit after eating it, they would feed the vomit to a dog, and if the dog died, then that individual would expect to die as well. And of course they would think that the poison will work faster in a dog, again because the dog is much smaller, so the dosage would be much bigger. Today, it's still a little bit of trial and error, but in a lot more of a regulated, systematic methodology. Essentially, today we expose receptors, which is a very cold term for the organism itself. A little bit confusing maybe because a lot of toxicology also looks at the mechanism of action, which looks at receptors on the surface of cells. But in general, a receptor in toxicology is the organism. Okay. So expose animals to various doses of a chemical and essentially see if they die or not, right? So generally the endpoint measured was mortality. Of course, this point, we recognize that it's not just enough to know if a poison is going to kill us or not. There are a lot of other negative impacts that we also want to avoid. So we also want to see if these chemicals can produce a tumor, a deformity, some other kind of functional deficiency long before it produces mortality. Okay, so ultimately, one of the main goals of toxicology is to look for the threshold dose, right? Is at this dose at which the chemical is going to produce these measurable effects, right? We can actually see them, measure them, recognize them. And of course, they are detrimental, right? Because chemicals can produce good effects, bad effects. We're looking specifically for the bad effects. So the threshold dose, of course, in these experiments, scientists choose which doses to expose the receptor to. And so at this point, we're looking for the threshold dose, but what we can actually measure is the lowest observed adverse effect level. So the lowest dose at which we actually see something bad happening. All right, chances are the threshold dose is somewhere around there or a little bit lower. The lowest than the LOAEL. Right? Lots of alphabet soup in toxicology. Okay. And of course, any exposure, any dose above this level you would expect to see an increasing prevalence of whatever endpoint you're measuring. So mortality, tumors, whatever effect you're actually looking at. And so the central concept in toxicology is this dose-response relationship, which can be expressed by this dose-response curve. So essentially graphically representing how much of a dose produces a certain likelihood of whatever the endpoint is we're measuring. And so what we can see here is the dose on the x-axis, increasing dose, so a bigger and bigger and bigger candy bar. And sometimes there is a time component as well, depending on the study, if it's just an acute toxicity test or a subacute toxicity test, etc. On the y-axis, we see the probability of response. Again, generally, what's measured here is mortality. Historically, it's been mortality, although in the last 50 years or so, a lot of other endpoints have been included in these experiments. Okay, so what we can see here is that essentially at these doses here, nobody dies, right? But when you start to give higher doses like these over here, you start to see a greater and greater and greater mortality rate, right? And so much so that at this dosage, any receptor, any organism that is exposed to this much of whatever we're talking about, they can all be expected to die. Okay. Other data points that these studies are interested in examining is the no observable adverse effect level. So for example, if the study uses a dose of 10, uses a dose of 50 and 100 and so on, the 10 level, as we can see here, there was no adverse effect level. Okay, so 0% mortality when the receptors were exposed to this this chemical. And so this would be the no observable adverse effect level. The next dose up would be 50. And so there we see there was some mortality. Okay, so that would be the lowest observable adverse effect level. So the threshold is somewhere between the lowest and the no. We don't know where, right? But to be safe, we're going to recommend this dosage is safe. Okay. Another data point that we are looking for is the lethal dose 50 or LD50. Essentially, the lethal dose, is the lowest dose to kill an animal during the observation period. So what is the dose here? We can refer to the LD1 or 50 or 100 to essentially express the lowest dose that kills 1% of the population, the lowest dose that kills half, the lowest dose that kills 100. Generally, the information that is relayed about chemicals is in terms of the LD50, right? So as we can see here, 50% of the population was killed by exposure to this much of whatever poison we're testing. Okay, so the LD50 or the median lethal dose. And for years, this has been used to essentially compare different chemicals, right? A lower LD50 means that chemical is more toxic than a higher LD50. We'll look at some examples here soon. So this has been the gold standard since like the 1930s or so. But this, of course, has no bearing on chronic exposure, right? Most of these studies are looking at acute exposure. But most chemicals, we might be exposed to for a lot longer periods of time. Also, if we're just looking at mortality, we can't really express anything about whether there is reproductive hazard or cancer risk or any others, any other impacts. So I wanted to come back around to chocolate. So chocolate is poisonous, right? Much to our chagrin. In the plant itself, and this is the species name here, there are a couple different chemicals that can act as poison. Caffeine is technically acting as a poison, and in future videos, we'll talk all about caffeine. Luckily, we generally consume a low enough dose that it doesn't affect us too much. Good thing, at least not as a poison. Theobromine, also called xanthiose, this chemical is what is actually able to disrupt the nervous system, the respiratory system, the cardiovascular system, and this is the one that makes chocolate lethal to your dogs. So essentially, this chemical can, in the short term, produce nausea and vomiting, diarrhea, lots of urination. So if you start to see these symptoms in your dog, you might suspect that they have gotten into your Snickers bars. Keep that in mind. Halloween is coming. What can happen if the dose is high enough, that is, the dog ate enough chocolate, is cardiac arrhythmias, epileptic seizures, internal bleeding, heart attacks, and ultimately death. So definitely want to keep your puppy away from this poison. So the LD50, right, the gold standard here. In humans, the LD50 of chocolate is about 1,000 milligrams per kilogram. And so what that translates to is you would have to eat about 12.6 pounds of dark chocolate or over 88 pounds of milk chocolate. If you're like 150-pound individual, right? So really a lot of chocolate. This is really not that toxic to humans. Dogs, on the other hand, we can see that the LD50 is much lower, and so it would take much less chocolate to produce all of these terrible symptoms in a dog. First of all, because they have a lower LD50, and this is because they don't have enzymes for this chemical that can break it down as quickly as we can. And generally, dogs are smaller than humans. So, eating the same amount, right, the same amount of chocolate in humans and dogs, it would affect the dogs a lot more because the dogs have a smaller body size. Right? So the dosage would be different. Might not think about this much, but cats actually have an even lower LD50 than dogs do. So what this means is that cats, or the toxicity is higher in cats than it is in dogs. But generally, we don't hear much about cats being poisoned by chocolate because the cats can't taste the sweetness in the chocolate like dogs can. And so generally, cats would not be inclined to eat as much chocolate to poison themselves as dogs would, right? Dogs find it very tasty. Just a couple other examples here. Note that this is the oral LD50 in rats. Okay, so oral, the exposure route, how toxic it is, and what is the organism. And so some substances which we would consider to be poisons, like strychnine here, is super toxic. The LD50 is less than 5, so very, very small dose would kill half the rats. But then other substances like opium, they can still be extremely toxic. You can see there's a range here. Very toxic, all the way down to practically non-toxic. Note that alcohol, ethanol, is slightly toxic to the rats. And so again, we can compare these LD50 values to get a pretty good idea of just how toxic something is. A little bit more, or more examples here. Here we can see, Botox, for example, is super toxic, right? Human oral. So very, very small amounts of Botox can actually kill a human. So botulism toxin from a bacteria. Of course, this chemical is used pharmaceutically for, not only cosmetic reasons, but also for other medical procedures as well. And so we can see here, kind of increasing LD50s. Most of these values are determined in rats. Okay, so we're going to talk about that here in a couple minutes. Just how do we get these values from any organism? let's see. Also, I want to point out that even things like alcohol, sugar, right, blood sugar, table sugar, water, we can also calculate the LD50 values for these as well. Um, and so once again, going back to Paracelsus' statement, in all things, there are poison. Everything has poisonous qualities. You can literally die from drinking too much water. In fact, in 2007, there was a radio contest called, something about win a wee for your wee, and essentially the contestants had to drink as much water as they could without going to the bathroom, and whoever could drink the most and hold it the longest would win a wee, which is a huge deal in 2007. But one of the contestants actually died from water poisoning, right? You can literally drink too much water and die, right? It's a lot of water. So don't worry. You should drink your water, by the way. Right. So just a couple examples here. What can we conclude about the toxicity of these three compounds? The details you're looking for here. Essentially is the LD50. So here is about 50%, with a tumor, so that, toxicity of 50%. So here, this particular compound produces 50% tumor rate with the dosage of maybe like 6 or 7, as opposed to number 3, which never produces a tumor in 50% of the rats. Okay, so what we can conclude about this is that compound number 1 is the most toxic and compound number 3 is the least toxic. Another example here, this one is borrowed from this paper right here, testing the effects of this poison. And so what can we conclude here? Well, what we can see is that the data has been divided into male and female. The LD50 curve, right, mortality, so the dose response curve is a little bit farther to the left in this example for males, for females are red, than it is for males. So the male curve is a little bit to the right. And so what that translates to is that the LD50 would be lower in females than it is for males. So this particular compound is more toxic to females than it is to males. Right? So when you're looking at these things, just look at where the curve is. Farther left is more toxic, farther right is less toxic. And so while we're here, I just want to make a brief side note about pharmacology. A lot of the methodology is the same, or at least overlapping, between pharmacology and toxicology. The reason for that, of course, is that the dose makes the poison. Sometimes, right, a little bit of ibuprofen, good. A lot of ibuprofen, toxic, lethal, right? So pharmacology and toxicology, are oftentimes studying the same chemicals. They share a lot of the same principles. So, how exactly the body handles exposure to these chemicals, it's going to take it up and excrete it in the same ways. What it's doing in the body is going to be the same, regardless of the dose, the principles of treatment, the dose response relationship. So a lot of toxicology is the same as pharmacology. And again, therapeutic drugs and essential nutrients, right, even things like water, can become poisons under certain conditions, particularly when the dose is high. And so, again, in toxicology, you're trying to produce these dose response curves. And we're generally looking for the LD50, the lethal dose 50%. So the dose at which it kills half of the individuals. In toxicology, you're generally looking for the ED50. It's the effective dose. When, or we're looking for how much ibuprofen should individuals take, or if we want half of the population to experience the positive effects, right? So effective dose 50%. So think LD50, but helpful. So these curves help us to establish what's called the therapeutic range. So we would want to prescribe, a dosage in this general range so that most people will actually get the benefits of this drug. Kind of between the effective range and the lethal range is the toxic range. So this is where, the ibuprofen would be causing, negative impacts, less than mortality, but still negative impacts, right? So negative side effects. And so it is important for a drug or for any substance to know the therapeutic range, right? Where is it, or at what dose is it helpful? At what dose is it toxic? And at what dose is it lethal? So one of the ranges that has been calculated and has some support and some opposition is the therapeutic index. And so that is essentially a ratio between the toxic dose of 50% and the effective dose of 50%. And so this gives us a standardized value for knowing just how safe the drug is. Right, so bigger values, so bigger difference between the toxic and the effective doses is better, right? That is safer because essentially there would be a huge difference between the toxic levels of the dose and the effective levels of the dose. And so this therapeutic index can be calculated, and it can inform physicians of, essentially how often do you need to, for example, get blood work in your patients to monitor, what is going on? We don't want them to, experience these toxic effects. So we need to monitor A, B, and C in their blood. Okay. Also the dosing interval. So are you going to take a pill at a high dose once a day, or are you going to take 4 pills at a lower dose over the course of the days, or over the course of the day? Right, so we can use these numbers to, most effectively treat patients with pharmaceuticals. Okay. So toxicity testing. In future videos, I'm going to talk about the pharmaceutical development process a little bit, but for now, I just want to talk about general household and commercial products. In general, manufacturers are required to test the toxicity of the chemicals that are in their products. So things like household cleaners, like chemicals that would be used in the laboratory, or in the workplace, and like agricultural chemicals and industrial chemicals as well. So there are laws that require manufacturers of a certain size. So it's like 12,000 pounds of, whatever every year, and grossing a certain amount of money, like $25 million or something like that every year. So big enough chemical manufacturers are required to test their own products and submit reports on the toxicity to various government entities. So for example, the EPA or the USDA monitor different types of chemicals, right? So the USDA might monitor Roundup, but the EPA might monitor household chemicals. Okay, so the companies must test their own products. And so this might send up some red flags or not for you. Right, so this is putting a lot on the company. The company decides how they're going to test. They decide, how long they're going to test, and ultimately that information is what the government uses to say, yep, that's good to go or not. So there is no objective outside source here. It's not peer reviewed or anything. This is all happening in-house, which of course, does not put the burden of testing on the government or any other outside entities. So this might save money, it might save time, but it is asking the individuals who are set to benefit from selling these products to, kind of be honest about whether or not they're toxic. So that is the topic that we're going to explore in a lot of detail in future videos as well. So just a thought for you, what would be the most convincing way to acquire toxicity data? So what data would you trust the most, right? Is this chemical okay to be used by humans? Okay, and so generally you might say humans, right? Like, is it safe in humans? We should know with some human data. Okay, unfortunately, human testing is wildly unethical, right? It is unethical to expose humans to a harmful substance on purpose, right? We're going to give these guys over here a lot. We're going to give these guys over here a medium amount, and these guys over here a little amount. That's just not okay. So where do we acquire some of this information about toxicity? A lot of information comes from observational data, from chemicals that are already in circulation. So sometimes, or in some substances, people can report, I've been exposed to this amount of a chemical and this is what I experienced. Okay. A lot of these observational studies are like substance use studies. So here we can see alcohol in the blood, right? So lots of people are exposing themselves to different amounts of alcohol. And so we can observe this guy drank this much alcohol and experienced this. This guy drank this much alcohol and experienced that. And so over many years, we can start to make these correlational conclusions. Unfortunately, there are drawbacks to this. Yeah, so there are drawbacks to that. First and foremost, observational studies can provide correlational conclusions, right? When this happens, we saw this happen over here, but because there are so many other variables, because the individuals are also eating cheese, they're also eating peanuts at the same time, maybe the cheese and peanuts together are what is producing this effect. Right, so it takes a lot longer and a lot more to actually make causational conclusions. You can also monitor like after an exposure or during an exposure. So for example, after 3 Mile Island, anybody who was in the area after that meltdown, you can study them and look for impacts of that exposure. Also after dropping atomic bombs in Japan, a lot of those individuals who were exposed and survived have been monitored over the decades to see what the long-term effects of those events might be, of those exposures. Also in the workplace, and even just like living in different types of environments. So for example, a lot of observational studies have been done on farm workers because they're exposed to so many different types of chemicals, and you can monitor different endpoints and compare that to individuals who don't work in an agricultural setting and see the correlation between them. Of course, there are not opportunities. There's not simply data available for every single chemical that is produced in part of our household or workplace products. And so generally what manufacturers do in order to test for the toxicity of their products is by using model organisms. Initially, primates were the go-to model organism because they are closest to humans. Of course, if the best data comes from humans, because of course, we want to know what's going to happen to us when we are exposed to these poisons, then primates are the next best thing because they are the closest. Of course, primates are not used generally for most toxicity testing now for a lot of different reasons. First of all, there were huge ethical concerns over using primates. They were very expensive to keep. The timeline for testing using primates was a lot longer because their lifespans were a lot longer. And many other downsides to using them as model organisms. Now, what is used or what are used most are mice because they have a very short lifespan, short gestational periods, so like 3 weeks or so. The They make a lot more of themselves very quickly, so they're relatively cheap model organisms, and we know a lot more about their genetics and their physiology, and we can compare this physiological process in the mouse, and it is similar in these ways to humans, and so we can kind of extrapolate data acquired through mice to the human side. Similarly, there are genetic lines of mice that we can use. There is limited genetic variation, and so the data might be more robust because there's less variability. And also, you can knock out different genes. So you can make it so the mouse doesn't express this gene, it expresses this other variation. And so we can test, how maybe this product affects individuals, that have this condition versus this condition just by manipulating the genetics of the mice, which, has been done for a very long time and is very well understood at this point. All right, so mice, very valuable and common model organism. Another very common model organism are the Sprague-Dawley rats. So your standard white lab rat that you see in all sorts of movies. They have similar benefits to mice. They also they have like a 2-year lifespan, so you can get some more long-term data out of them. There are some other model organisms that have been used for various toxicity testing, although they are surely at this point much less common than rodents. Most or a lot of toxicity testing has used model organisms in vivo or in life. Living organisms are exposed to these different chemicals. There is no gold standard, absolute method for toxicity testing using these different organisms, but in general, this chart is showing us what most places do. So for example, to test acute lethality, like does this chemical kill after one exposure or an exposure over the course of a single day. Generally, places use one species, for the most part, rodents or rabbits. Generally, they have 3 or more individuals. And so I just want to point out, like, is this chemical going to kill people? 3 mice, did they die or not? No, they didn't die, therefore it's good to go. Right, so small number of animals, maybe that's good, maybe it's not, we'll talk about it in a second. Route of administration, so this may or may not be comparable to how humans would be exposed, so it might be orally administered, but maybe in reality, humans would be exposed to it via dermal exposure. Okay. Animals are monitored for a week or 2, and pretty much are they dead or are they living after that time? And so similar methods for all the way through chronic toxicity. So I encourage you to think about the benefits of testing with animals and the disadvantages of testing with animals. There are many of both, and surely you could argue in both directions. I have just a couple examples here of some advantages and disadvantages that I'll read through for you. So for advantages, generally it is considered to be more ethical to experiment on animals than on humans, again, because it is not acceptable to expose humans to different amounts of chemicals, which you are pretty sure have toxic effects. For example, if Clorox comes out with a new product to clean your shower, clean your tile or something, it is possible that that chemical could splash up into a human's eyes and therefore harm them. And so Clorox is required to test on that product, or to test the toxicity of that product. Is it going to hurt the human's eyes? And so if we use human testing, it would require humans to go in and actually get this product splashed into their eyes and then see what happens. And so generally, that is not okay. And so animals are used instead. We'll talk more about that here soon. Experimental conditions can be carefully defined and controlled. And so we can actually make causational conclusions when an individual is exposed to this. These are the things that can happen, as opposed to having so many other variables that are complicating your conclusions. You can use a specific strain of rodents where their genetic line, and you can reduce the genetic variability. And a lot of different information can be obtained from using animals. A lot of times the animals are euthanized at the end of the experiment, and you can conduct histological and biochemical analyses to see exactly what is happening with exposure to different poisons. Surely using rodents is relatively inexpensive, and so that is much more appealing to different companies, right, or labs anywhere. Disadvantages to using animal studies. Oftentimes these studies are only looking at acute toxicity, and so the more long-term consequences are, we don't know the long-term consequences. So rodents have a lot in common with us. A lot of the metabolic pathways are the same, if not very close. And so we can extrapolate data from rodents and make conclusions about humans with a reasonable level of confidence. However, that is not always the case. Just one example here, nickel-induced contact dermatitis. Nickel has no impacts on rodents in the studies, but in human cells, there is a risk of this particular condition. So not all of the pathways are the same. And so sometimes, you can make pretty robust conclusions in rodent models, but the products get out into circulation and humans have a very different response. Of course, probably the first thing that you might think of as a disadvantage to animal studies is that, yes, maybe animal experimentation can be considered by many to be more ethical than experimenting on humans, but is it actually ethical? Especially when you're looking for adverse health effects, you are looking for the level at which half of your population is killed by this chemical. And so the point of these experiments is to cause adverse health effects and mortality. So is that ethical? And finally, because there are so many variables, so many different processes occurring within a living organism in vivo, sometimes it is difficult or impossible to determine the actual mechanism of the damage or the mechanism of the disease that is caused by these poisons. And so, without knowing exactly how it works, we can't always fix the product. We can't always, you know, improve the methods, without this deeper understanding. And so, more and more toxicity testing is moving away from animal models, for better or worse, is moving away from animal models. And part of that drive is due to the Animal Welfare Act, which demands that there is a reduction, a refinement, and a replacement of animal testing as much as possible. So reduce the number of animals, reduce the number of tests, refine the methodology so you are not causing as much harm to as many organisms, and when possible, replace animal studies with something else. So this is an image which shows, that, using animals is questionable for a lot of the reasons I mentioned and more. And so what might be a better alternative is to use in vitro technology. So essentially to get tissue from various sources. So the skin, the eye, the kidney, the brain, the liver, for example. And so you can use tissue culture from any of these. You can actually take a slice of an organ. Right? So you actually have a little bit more tissue and have some more of the complex interactions that are occurring within tissues as opposed to isolated cells. And there can be some cell lines that are actually developed to test these more specific questions. And so developing these and using them for toxicity testing, that should be the goal, right? Of course, it is quite a process to develop these sources, but ultimately it takes away the need for at least as many animals. So ideally, human cell culture would be best because, of course, we want to know what the human response is going to be, not, a random rat. This is challenging because there is an inconsistent supply of tissue, specifically or especially things like brain tissue, right? There's only so much brain tissue that you have, and usually like donor tissue, might be older tissue or might be like only certain demographics or genetics. And so this might not reflect all of the possible reactions to different chemicals. Of course, there's a lot of variability between donors. For example, skin variability is quite significant. Cells may not express mechanisms that are required for the compound to be toxic. So yes, individual cells might not show an impact, but when those cells are actually in the liver and they're receiving constant supply of substances from the blood and from the digestive system, then maybe the compound can be toxic, right? There's a lot of different factors affecting toxicity, which you are eliminating a lot of those factors, right, by using just cells. For example, the microenvironment might impact the response to chemicals, cell-to-cell interaction. So in culture, maybe you're looking at one specific cell type, but maybe this cell and another type of cell might interact in such a way that makes the product even more toxic. So in addition to human cell culture, Another tool which is continually being developed and updated are chemical databases. So essentially these are repositories of information from a lot of different entities, which kind of categorize chemicals, this general shape or this general type of molecule has been shown to produce these effects. These are the mechanisms. These are the impacts on this type of tissue and this type of tissue. And so even before you actually conduct these experiments, you can predict what mechanisms are going to be at play, and therefore you can design your experiments to be the most effective, right? You already know kind of what you're looking for. There's also been a lot of work in genomics, proteomics, metabolomics, so looking at genes, and maybe this particular genotype is affected in this way, and this particular genotype is affected in this way. And so there's information from previous studies that can help inform what this chemical's impacts are going to be. There have been certain gene clusters that have been identified for things like cancer, or different types of cancer, breast cancer, etc. And so there's already a framework for understanding how different types of chemicals are going to affect cells of this genetic composition. Also, a lot of information on proteins, on metabolic pathways. And so the more information is accumulated from labs throughout the world, both commercial labs as well as government and higher education labs, the more basis for understanding there is to develop studies for new products. And so there is, finally, right, all of this information can be processed with some advanced computer modeling, again, predicting the effects before you actually do these tests. And so you can pick exactly the right type of cell culture, exactly the right type of experiment to, again, reduce the need for animals as much as possible, of course. So most of this testing that I've been talking about is, like chemicals that you would use in a lab or in like the cleaning industry or agricultural industry, and just like common household products. The testing for cosmetics and drugs that are going to be used to treat eye conditions, so things like eye drops, and like the drops they use at the eye doctors to dilate your eyes and everything, those types of things have a particular type of testing that is historically been used for them. And the reason for that is that in the early part of the 20th century, a lot of new products coming out, a lot of new cosmetic products, such as in this example here, a type of mascara. And at the time, there were no strong and enforced regulations on how much testing and what type of testing had to be done for cosmetic products, etc. And so what started to happen was that these new products started to produce lots and lots of toxic effects. So this is actually a flyer that was made summarizing what this mascara did to this woman here. Here's what she looked like before, and then after using this mascara, here's what her eyes looked like. She and others like her, she's just the one example here, developed all sorts of different conditions, and ultimately this led to her blindness. And so in the early 1930s, there was an outrage over this. Like, people were not comfortable. They couldn't trust that products weren't going to make them blind. And so essentially, they went to the Senate and said, we need better regulations, we need more monitoring of cosmetic products, etc. So just like the LD50 testing, which was developed in in the 1920s or so, the Dray's eye test, developed in 1944, was meant to standardize methodology. Like, this is the test that is done so that we can compare the toxicity of different chemicals, and we can present this information to individuals who are going to be affected, or to government entities that regulate these products. And so anything that could have accidental contact with human eyes, right? Again, like the tub and tile cleaner by Clorox, like that could splash in your eyes, that would have been tested with the Dray's eye test. Also, of course, cosmetics and eye care products, of course, are meant to be, around the eyes. And so what is the Dray's eye test? The Dray's eye test is an in vivo test. That is, it is using live animals to assess acute ocular toxicity. So toxicity in the short term to the eyes. And so generally the model organisms that are used are rabbits because they have really big eyes. And so it's relatively easy to see the effects of the products. And this is, what a healthy rabbit eye would look like. Okay, so what this test requires is the injection of a small amount, a standardized small amount of this test liquid, whatever you're testing, the mascara or the Clorox, whatever, into the lower conjunctival cul-de-sac. So essentially it's to like pull the lower eyelid back and stick it right under, under the eyelid, and then rinse it out after a certain amount of time. Observations would proceed after an hour, after a day, a couple days, then ultimately a few weeks out. And so just these observations would make note of any inflammation, any of those conditions that were listed on the previous slide, etc. At the end of the observation period, after a few weeks, the animals with irreparable damage to their eyes would be euthanized and studied in more detail. And of course, euthanized for ethical reasons, right? So to not let the animal suffer any longer. Through most of the 20th century, this was the method for testing all of these chemicals that I listed before. Very, very common test. Today, it is criticized for the fact that there isn't some kind of objective system for quantifying exactly what the effects on the eyes are. It's just looking at it and, is this happening? Is that happening? And then, making notes. Also, this is unreliable for predicting more long-term effects. The observations are only made for a few weeks. What's happening after that? And of course, animal welfare groups have been very vocal about these types of tests because of the potential suffering or potential major suffering of these animals. And so many companies have actually made statements, and committed to not using this type of testing because it is unethical, because it is not dependable enough. And in fact, there are lists by a few different animal welfare organizations that list companies that do not test on animals for their cosmetic products. Of course, there is not necessarily a standardized alternative to this test at this point. And so, there is an opening, an opportunity for developing a more ethical and a more dependable test for commercial products moving forward. Okay, so thank you very much for listening to this video. The next video is going to be all about how exactly poisons work in the body, right? Actually looking at the mechanisms of action and how your body processes them. Thanks so much.